So in genetics we will be considering the two major concepts. One is the gene frequency and the second one is genotypic frequency. These two will be important because these will determine whether the population exists in equilibrium or not. For a population to exist in equilibrium, the gene and genotypic frequencies should be constant. So from one generation to the next generation, if you go on calculating the gene and genotypic frequencies of a population which is present in the equilibrium, then they remain constant. Such populations cannot be subjected to evolution. For a population to be subjected to evolution, it is a must that the population is subjected to evolutionary forces such as mutation, migration, genetic drift, and random mating. So when you have this kind of events, then there is a possibility that the population can evolve. The gene frequency is called allele frequency. Which means the proportion of an allele in a given population. That is, the number of individuals having that allele divided by the total number of alleles, total number of individuals. The genotypic frequency, the same way, the proportion of a given genotype in population. So you take that genotype and then you find out the total population. From that you find out the proportion of the genotype. To understand these two concepts, we take a simple case where, let us say, we have a gene which is diallelic. So when you say a gene is diallelic, it means it has two contrasting copies or two contrasting alleles. So for simplicity we will say one allele is dominant and another allele is recessive. So you can have a case, maybe they are co-dominant. You can also have a case where one is partly dominant, another one is recessive. But to make the things easy, we are taking one allele as dominant, one allele as recessive. Now for this gene, when you look at the genotypes, you can get three possible genotypes. The first one, homozygous dominant. The second one, homozygous recessive. The third one, heterozygous. So for a gene having two alleles, the maximum number of genotypes that are possible will be three: homozygous dominant, homozygous recessive, and heterozygous. Since we are looking at population, remember, each individual is dijoking. Each individual is diploid. What does it mean is contains two alleles for a locus or for a gene. So one individual may have capital A for that particular locus or position. Another individual can have small a for that particular position. Another individual can have capital A small a for that particular position. If I look at the frequency of capital A, f of A, I can represent this with a symbol, the usual symbol that we take for the first allele is P. This will be the number of individuals having capital A, because each capital A has two alleles, 2 times, plus number of individuals having capital A small a. Here we have to multiply with 1 because there is only one capital A. We avoid that. We simply say number of capital A small a individuals divided by total number of individuals. Again, we said each individual has 2 alleles. So it is 2 into total number of individuals. Or if you make a relatively simple formula for exam purpose, number of homozygotes plus half the number of heterozygotes divided by total number of individuals. So this will give us the frequency of dominant allele, which we have considered as P. The same way we can go for frequency of the recessive allele. Now we take the next letter in the alphabet, which is Q. Again, 2 times the number of homozygous A individuals, AA, because each one has 2 alleles, plus the number of heterozygotic individuals, since each one has only one small a, divided by each individual has 2 alleles, so 2 times total number of individuals. This again we can simplify as number of homozygotes plus half the number of heterozygotes divided by total number of individuals. So that gives us gene or allele frequency. Now we have to find out genotypic frequency. So we need three genotypes. Capital A, capital A, capital A, small a, small a, small a. So to find out the genotypic frequency, we make a checkerboard. So one side of the checkerboard we take the female gametes, another side we take the male gamete. This is the general convention. Here, frequency of dominant allele, already we found out P. Frequency of recessive allele, we find out as Q. Frequency of dominant allele, we assigned P. Frequency of recessive allele, we assigned Q. So when the gametes combination, we consider this is a random combination. Any gamete has equal opportunity to combine with the gamete of other sex. So one combination is these two. So P times P is P square. The second one is this. That is P times Q, which is PQ. Third one, frequency of capital A, small a. Again it is PQ. And finally, frequency of small a, which is Q square. So now we got the genotypic frequencies. So frequency of capital A equals to this column, which is P square. Frequency of small a equals to this column, which is Q square. Then frequency of capital A, small a. Here we should be careful because capital A, small a occurs at two different places. One here and one here. So we got PQ plus PQ. Equals to 2PQ. So these are genotypic frequencies. So we have gene frequencies, how to calculate, and genotypic frequencies, how we can calculate. The next thing with respect to these frequencies is the sum total of frequencies should be equal to 1. So if I take sum total of allele frequencies or gene frequencies, which is P plus Q, it should be equal to 1. Since we have two alleles, and the allelic frequency should be equal to 1. If you have 3 alleles, then the sum total of 3 allele frequency should be equal to 1. If you have 4 alleles, the sum total of 4 allelic frequency should be 1. You can't have frequency that is greater than 1. Since P plus Q equals to 1, I write P equals to 1 minus Q. Similarly, I can write Q equals to 1 minus P. So if I have given the frequency of dominant allele equals to 0.3, find out the frequency of recessive allele. So dominant allele, that is P, so 1 minus P. Q equals to 1 minus 0.3, which is 0.7. So now we have the sum total of the two alleles equals to 1. This is how you do when you are given one frequency and then you are asked to find out the second allele frequency, or even you can calculate the genotypic frequencies. The same rule, that is sum total of frequencies is equal to 1, we can also apply for genotypic frequencies. So sum total of genotypic frequencies is also 1. That means P square plus Q square plus 2PQ equals to 1. If I know two frequencies, I can find out the other frequency. P square will be, so transfer this to the other side, 1 minus Q square plus 2PQ. Similarly, if you want Q square, 1 minus, transfer this to the other side, P square plus 2PQ. You want 2PQ, again the same. So at gene frequency level, sum total of the frequency is 1. At genotypic level, the sum total of the frequency will be 1. Now in populations, what basically happens is, as the frequency of one allele increases, the frequency of another allele decreases. So let us start a case where frequency of capital A is 0. And then we go gradually where frequency of capital A will become 1. That means here we have only small allele, here we have only dominant allele. So here it will become frequency of small allele equals to 1. Here frequency of small allele equals to 1. So what happens? You have gradual increase in the frequency of the dominant allele. So this is actually the genotypic frequency. You can write frequency of capital A equals to P square. So P square gradually increasing and becoming 100%. If you take the second side, the frequency of small allele goes on decreasing and ultimately it reaches 0. So frequency of small a equals to 0 here. So that in terms of factor we give Q square. Then what about the frequency of heterozygotes? So let us say this is a 50% mark. This is what happens to your frequency of heterozygotes. So frequency of AA equals to 2p. It's a very interesting frequency distribution because heterozygous frequency, the maximum that we get is 0.5 or 50%. In any population which is equilibrium, you cannot get heterozygous frequency greater than 50%. So heterozygous frequency is gradually increasing, reaching 50% and then gradually decreasing, again coming back to the 0 level. So this is a unique feature in terms of the frequency distributions. If you take here this point, here P will be equals to, Q will be equals to 0.5. And it doesn't matter whether it is P square or Q square, this will be 0.25. So when the frequencies of the two alleles in a population, when they are equal, their genotypic frequency will be equal to 0.25. One important aspect. Second aspect, when an allele is low in concentration, when allelic frequency is low, most of that allele occurs in heterozygotes, not in homozygotes. When the frequency of one allele increases, the frequency of another allele decreases. When the frequency of one allele increases, the heterozygote frequency increases, reaches a maximum level of 50%, after which it decreases. So any population where heterozygous frequency is more than 50%, you can consider that as a non-equilibrium population. It cannot exist in equilibrium. This population is undergoing evolutionary changes. So we'll see a simple problem to understand the whole concept. So let's say in a population in equilibrium, so we are giving the condition beginning itself, the frequency of dominant allele is 1%. Calculate the frequencies of various genotypes. So you are given one frequency, and that too the P is given, dominant allele. So dominant allele frequency can be anywhere from 1% to 100%. Recessive one can be anywhere from 1 to 100%. So it doesn't bother about that number. Since they have given as 1%, we will take it as 1%. So frequency of dominant allele equals to P equals to 1%. This we can write it as 1 over 100. That means 0.01. Then the frequency of recessive allele will be Q, and that will be 1 minus P. So 1 minus 0.01, that means it is 0.99. Now if you want the frequency of capital A, it will become P square, which is 0.01 whole square. If you want frequency of small a, it will become Q square, which is 0.99 whole square. If you have frequency of heterozygotes, it will become 2 into 0.01 multiplied by 0.99. The sum of all this will be equal to 1. So that's how you find out the frequency from allelic frequency. Here remember, since we are given only P, there is only one way we can find out Q, that is Q equals to 1 minus